You are listening audio paper series with Yasir, and today we will discuss a recent publication of Dr. Keith Turner, Genome-Wide Analysis of Innate Susceptibility Mechanisms of E. coli to Colistin. It is published in MDPI journal Antibiotics. Welcome, Keith. Why did you study E. coli susceptibility to colistin?
Hello, Yasir. Thank you, and thank you for having me. Why colistin? So, colistin is an antibiotic that is being used more frequently to treat bacterial infections, largely because of the emergence of antibiotic resistance towards so many of the antibiotics that are currently available to us. So it's effectively being used as an antibiotic of last resort, unfortunately. And if we can understand how colistin works, then it provides us with opportunities to render resistant bacteria sensitive to colistin once again.
Okay, so how did you study these susceptibility mechanisms?
So we used a whole genome assay that we've developed at the Quadram Institute, which we call TRADIS Express, and this allowed us to assay every gene in the bacterial genome to see which of those genes are involved in sensitivity or resistance to colistin. And the way this works is that we generate a very large number of mutants of the bacterium of interest, such that we will have maybe dozens or maybe even hundreds of different mutants representing each gene. And our assay is able to determine which of those mutants increase in numbers and which decrease in numbers when we grow all these mutants together in a mixture that contains a small amount of colistin. And therefore any genes that are involved in resistance will increase in numbers, and those involved in sensitivity will reduce in numbers. And we can measure those differences.
So you used TRADIS Express to study colistin susceptibility mechanisms. What are the major findings of this study?
Well, we found a wide range of genes that are involved in colistin susceptibility, and these included genes that code for the biosynthesis of lipopolysaccharide and other membrane-associated proteins. LPS, of course, is the very outer surface of the bacterial cell. We also found some genes that are involved in transcription and translation. So these are genes that are actually involved in gene expression. We found genes coding for proteins that transport molecules across membranes. We also found genes that are involved in DNA metabolism, such as helicases and DNA repair proteins. And in addition, we found some genes involved that are of previously unknown function. So now we can start understanding what these genes may be, what their role may be in the bacterial cell.
Okay, so there are quite few mechanisms are involved, but the major contribution is coming from cell envelope.
Yes, that seems to be the case, yes.
Thank you. So what is the significance of this study?
Well, now that we've screened the whole genome, we know not only which genes are involved in colistin susceptibility, but we also know that all those other genes are not involved. And this provides us with clues as to how colistin affects bacterial cells as a whole, rather than it just telling us specific things about the component of the bacterial cell that colistin itself targets. In addition, we see, for example, that inactivation of a gene called RECF confers tolerance to colistin. RecF is a DNA repair enzyme, and this allows us to predict how colistin resistance may develop in the future. Also, we see genes where inactivation increases susceptibility to colistin. So any compound that was able to target those gene-encoded cell components could return colistin-resistant bacteria back to sensitivity.
Thank you very much, Keith, for sharing very interesting paper.
Thank you, Yasir.